Hello. We got new microphones, that's good. Yeah, and end to the hiatus. Hello, everyone, and welcome back to Genetics Unsealed. I am your host, Following Adeleke, and today we'll be talking about optogenetics, optovolution, and the role of R in modern bioinformatics. Optogenetics is a biological technique that combines genetics and optics, the study of light. It allows scientists to control the activity of cells, especially neurons, using light. To understand this, remember a key idea from A-level biology. Neurons communicate through electrical signals generated by ion movement across membranes. Optogenetics works by inserting genes into cells that produce light-sensitive proteins, usually derived from microorganisms such as algae. One well-known example is chronorhodopsin, a protein that opens ion channels when exposed to blue light. When light shines on neurons containing these proteins, ion channels open, charged ions move into the neuron, and the neuron fires an electrical impulse. In simple terms, scientists can switch neurons on or off using light pulses. This allows researchers to study exactly how specific groups of neurons influence behavior. Before optogenetics, neuroscientists could observe brain activity, but struggled to prove cause and effect. For example, imagine a region of the brain become active when an animal feels fear. Is that region causing the fear, or is it simply responding to it? With optogenetics, researchers can activate these neurons artificially using using light. If the animal immediately shows fear behavior, scientists know these neurons play a causal role. Optogenetics has been used to study memory formation, reward pathways in addiction, sleep regulation, movement disorders like Parkinson's disease, and psychiatric conditions such as depression. Because scientists can target specific neurons genetically, the technique is incredibly precise compared with older electrical stimulation methods. Now, introducing a new development that builds on optogenetics, optoevolution. Researchers at the École Polytechnique Fédérale de Lausanne, EPFL, developed a light-guided method that allows proteins to evolve dynamic behaviors inside living cells. To understand why this is important, we need to revisit a key evolutionary concept, directed evolution. In laboratories, scientists often use directed evolution to engineer proteins. This works similar to artificial selection. Just as farmers breed crops or animals with desirable traits, scientists create many variants for a protein and select the ones that perform best. Directed evolution has produced useful proteins for medicine, industrial enzymes, biotechnology, and antibody therapies. However, traditional directed evolution has a limitation. It usually selects proteins that are constantly active, but many biological proteins behave differently. They act like switches. They turn on briefly, then off, then on again, depending on signals inside the cell. Existing evolution techniques struggle to create proteins with these dynamic behaviors. The EPFL researchers solved this problem using light-controlled evolution, which they called optovolution. Their system used the yeast Saccharomyces cerevisiae, a species commonly used in baking, brewing, and biological research. The scientists rewired the yeast cell cycle so that survival depended on whether a protein switched on and off at the correct time. If the protein stayed active for too long or inactive for too long, the yeast cell stopped dividing or the cell died. Only cells where the protein switched correctly could continue reproducing. Each yeast cell cycle, about 90 minutes, acted as a test of whether the proteins behaved properly. This allowed evolution to automatically select proteins with the correct switching behavior. To control the process precisely, researchers used optogenetics. They exposed the cells to time pulses of light, forcing proteins to switch states at specific moments. If a protein evolved the correct response to these signals, the yeast cell survived and divided. If not, the cell failed. Over many generations, evolution produced proteins that behaved exactly as needed. This method allowed researchers to evolve proteins that respond to different colors of light, switch on and off dynamically, and behave like biological logic gates. One of the most fascinating outcomes of optovolution was the creation of a single protein computer. The evolved protein activated genes only when the two signals occurred simultaneously, a light signal and a chemical signal. This is similar to an AND logic gate in computer science. In digital circuits, an AND gate only produces an output if both inputs are present. Biological systems often use similar logic to make decisions such as whether to divide, whether to activate immune responses, and whether to repair DNA. Optovolution therefore helps scientists design synthetic biological circuits. These could one day be used to engineer DNA, to engineer cells that detect disease, produce medicines inside the body, and or respond to environmental signals. The researchers also evolved new variants of optogenetic proteins. They discovered 19 new versions of a commonly used light-controlled transcription factor. These new proteins had improved properties, greater sensitivity to light, reduced activity in darkness, and responsiveness to green light, not just blue light. Using multiple light colors allows scientists to control different cellular systems independently, much like using separate remote controls. The team also evolved a red light system that no longer needed an extra chemical cofactor. Evolution produced a mutation that allowed the protein to use molecules already present inside the cell. This makes the system much easier to use experimentally. Experiments like experiments like optogenetics and optovolution generate huge datasets. Scientists might measure gene expression levels, protein activity, cell cycle behavior, neural firing patterns, and DNA mutations. This is where bioinformatics becomes essential. Bioinformatics combines biology, statistics, and computer science to analyze biological data. One of the most widely used tools for this is the programming language R. R is a programming language designed specifically for statistical analysis and data visualization. It is heavily used in genomics, neuroscience, and systems biology. R helps scientists analyze DNA sequencing data, identify gene expression patterns, build statistical models, and create graphs and visualizations. For example, when researchers study how genes respond to light signals in optogenetics experiments, they may collect thousands of measurements. R allows them to organize, analyze, and interpret that data efficiently. Several powerful R tools are used in biological research. For example, DSEC2 and EdgeR. These packages analyze RNA sequencing data to determine which genes are significantly activated or suppressed. ggplot2. This package creates high quality graphs and visualizations for scientific papers. Using R, scientists can detect genetic mutations, compare gene expression between healthy and diseased cells, model biological networks, and analyze neural activity patterns. In optogenetics experiments, R might be used to analyze neuron firing frequency, behavioral responses to stimulation, and gene activation after light exposure. When combined, optogenetics, optovolution, and bioinformatics represent an incredibly powerful research toolkit. They allow scientists to control biological systems of light, evolve proteins with complex behaviors, and analyze massive biological datasets. These technologies are opening new possibilities in neuroscience, synthetic biology, gene therapy, and personalized medicine. In the future, scientists may design programmable cells that detect disease signals and release treatments automatically. And as biological experiments generate ever larger datasets, programming languages like R and Python will become increasingly important for analyzing and interpreting the results. That's it for today. I hope you enjoyed this episode and the sound quality. This is your host, Follow me Aleshona Deleke, signing out.